Okay, so uh, up to the last lecture, I have already finished my lectures on my syllabus. That is, except the except the prodrug design, except the prodrug design, the rational drug design part and combinatorial chemistry, various methods of combinatorial synthesis, and your uh, then your various assay methods of combinatorial library, QSR model development, validation, different statistical methods, different models, Free Wilson, Topliss, then Fujita band, everything we finished. Then 2D QSR, 3D in 3D QSR we have discussed the various methods, 3D QSR, COMFA, all these things we finished. Actually, my portion is finished except the prodrug one. But now I am going to start a new one that is called computational prediction of protein structure. Computational prediction of protein structure. So this is in your syllabus. I think in module 2 or somewhere it is there. And I have been asked to finish this one on behalf of you. So I'm starting this one. That is the computational prediction of protein structure. Computational prediction of protein structure. Okay. What does it mean by computational prediction of protein structure? This is, if you recall, there in the drug design step I told, if you can go to structure-based drug design, if you know the target protein structure or target receptor structure or target enzyme structure, but you cannot go to the structure-based drug design if you don't know the target protein structure, right? But now we are finish the backgrounds of these, so we will now advance to the this step. That means you can do that. Now we can add up one thing, that we can do target specific or structure specific drug design even if we do not know the target protein structure. Even if you do not know the target protein structure, target protein or receptor structure, even if you do not know this, you can do the drug design. How? That is through the computational prediction of protein structure. That means you do not know the protein structure. If it is a target protein structure, you do not know. There is a question mark, means you do not know. But still you can do this by predicting. Now you can predict. Now you can predict the protein structure, and based on the predicted protein structure, now you can move on to the drug design. Then you can do the docking, virtual skidding, all these things, and you can move on to the drug design. Now this is the importance of predicting protein structures, because it has been, I will send you notes. That there are I will send you one paper from one journal, Indian Journal of Pharmaceutical Sciences. In the last half of that paper, you will see lots of drugs actually have come out based on this computational prediction of protein structure. That means there are several receptors in our body or several proteins in our body where people didn't know the actual structure. People didn't know the actual structure of that protein, but they discovered drugs based on that. How? Because they predicted the protein structures and then they discovered the drugs. Lots of drugs have been evolved like this. I'll send you the paper. In the last half, you will see one table. In the table, or there is one paragraph. In the paragraph, you will see that these drugs have come out like this. So first you have to know how we can predict the protein structure. Now, predicting computational prediction of protein structure, you can divide in 3 steps. The first step, okay, the second step, and the third step. Okay, third step. What are the steps? I first write down what are the steps. Okay, the first step is that is a mandate that we should know the sequence of the protein structure. That we should know the sequence of the protein structure. Okay. Next, should know the sequence of the protein structure. Next, we have to model the protein structure. We have to model the protein structure by a method called homology modeling. We have to model the protein structure by a method called homology modeling. Okay. And then we have to move to the third step. That is, we have to predict the accuracy and acceptability of the model. That is called model validation. Okay, we have to predict the accuracy and acceptability of the model. It is called model validation. So there are three basic steps. Okay, all the steps I can show you in practical. That means how the sequence of the protein structure is found. Starting from that, how we can model and how we can predict the accuracy and acceptability of model, everything can be shown in practice. Actually, in my drug discovery course, okay, the first section of this, that is deriving the sequence of the protein structure and some portion of the homology modeling is there, okay, in my drug discovery. But here in this lecture, I don't have that amount of time to go through all the practice session right now. If possible, I will later send you the videos where you see using the internet and using the different tools how people are doing this homology modeling. But before that, we have to know the theory first. If you don't know the theory, you cannot approach that computational prediction of protein structure. Okay, so first we have to know the sequence of the protein structure. Okay, so how you know the sequence of the protein structure is the first question. But before that I would like to highlight one thing. That is that we have to know a brief about protein structure. A brief about protein, because if we do not know a brief about protein structure, you cannot understand how the accuracy of the model depends or how the validation of the model depends. So protein structure, first we need to have a brief. Okay. Now protein structure, I will not start from the very beginning, because all of you know that protein are built up of amino acids. Okay. So amino acids portions, we all know proteins are built up of amino acids. These amino acids are linked. Okay. These amino acids are linked like that. How they are linked? Say, they link like, say, amino acid AA1, then AA2, okay, AA1, AA2, AA3. This way the amino acids are linked. Here your protein concept starts. The amino acids are linked like how they are linked. They are linked by a linkage called peptide linkage, that is CONH linkage. Okay, CONH linkage, that is called peptide linkage, or you can say NH-CO linkage. Through these peptide linkages, amino acids are bonded with each other. Fine. So you all know this. How this peptide linkage comes from carboxyl terminal of the previous amino acids to the amino terminal of the next amino acids. Fine. This we know. Now, from here, what is this structure now? This also describes the protein structure. What is the structure? This structure is primary structure of protein. This structure is primary structure of protein. Okay, because primary structure means you know the sequence of the amino acids, and from the sequence you have the idea of the protein, how it look like, or what it will be, something like that. Now after that comes to the secondary structure. Secondary structure, we know secondary structure is comes like alpha helix or beta sheet. So if there are helix goes along like that, there are hydrogen bonded, the helix if are hydrogen bonded like this, okay, then it is called alpha helix. It is called alpha helix. And if these alpha helix have a lot of hydrogen bond and there are a lot of water molecules inside, but if you take out these water molecules out, and make it a very robust dry structure, then the structure becomes something like this, that is called this. So this is pleated structure, sheet-like structure. It is like a staircase. It is either going down or going up. So this is pleated, a pleat-like structure or sheet-like structure. So these are called beta sheets. These are called beta sheets. We all know alpha helix and beta sheets. Okay, this alpha helix can be parallel, can be anti-parallel, depending on the hydrogen bond. Okay, if one alpha helix, if one strand comes from top to bottom and other strand also comes from top to bottom, then it is called parallel alpha helix. But if one strand comes from down to bottom and one strand comes from top to bottom, then it is called anti-parallel alpha helix. We know this. Okay, so alpha helix beta sheet, we know this is called secondary structure. This is called secondary structure. I'll write here. This is called secondary structure, right? Why secondary structure? Because here in primary structure we do not have any idea about the 3D conformation of the protein, no idea, only you know the amino acid sequence. But in secondary structure, although we do not have the full 3D conformation, we have the part of orientation of the protein, part of what I'll say, part of the going up or going down, or part of the orientation, or part of the alignment of the protein. These things we can have some idea. That's what is called secondary structure. In the tertiary structure, we come to the tertiary structure. Sorry, we come to the tertiary structure where we have the full 3D conformation of the protein. This is the actual structure where we have the 3D conformation of protein. That means the 3D orientation of all the protein backbones, residues, and the side chains. So this is the tertiary structure. This is the tertiary structure. Sorry, tertiary structure. And lastly, we come to the quaternary structure. That is very ionic, compact kind of structure. Sorry. This is very compact kind of structure. Quaternary structure is very compact because this is highly ionic. So they are very compact and concentrated on a small place because of the ionic interaction. So this is quaternary structure. This is quaternary structure. Quaternary structure. Okay. Apart from that, quaternary structure is a special structure of protein when protein is highly ionized in a particular fluid. Apart from that, all other protein conformation, if we want to have the actual 3D conformation of protein, we have to look at the tertiary structure. Normal protein structure means the normal 3-dimensional protein structure means your tertiary structure. Means your tertiary structure. This one. So tertiary structure gives you the actual all the informations, the 3D conformation, the actual spatial orientation about protein. So when I am telling that protein structure, we always unless otherwise stated, we will focus on the tertiary structure. Sometimes we focus on the alpha helix beta sheets, but always remember that the alpha helix or beta sheets are also involved in this tertiary structure. In the tertiary structure, there is also involvement of alpha helix beta sheet. So when you compute the tertiary structure, when you predict tertiary structure, already the portion of alpha helix and the portion of beta sheets are predicted. Okay. So now this is a very basic ideas of the structures. Now we know something about we know more about this protein structure. If we were to go for computational prediction, what is the more about protein structure? Now first thing is that we have to know about phi angle and psi angle. Phi and psi. I do not know if you have studied this previously or not, but now you have to come to this first, phi and psi. What is phi angle and what is psi angle? Phi angle is called torsional angle and psi angle is called dihedral angle. So phi is called torsional angle and psi is called dihedral angle. What is torsional angle and what is dihedral angle? Torsional angle, when there is a 3D conformation of protein, if you look at that, if you consider a particular segment of protein and you consider on a particular bond, say the protein bond is like this. Say the protein bond is like this. Say the protein bond is this, then this, something like protein bond is this. Now these protein bonds, if these are the atoms here, these bonds do not lie in the same plane of the atoms. These bonds do not lie in the same plane of atoms. Rather, it is somewhat distorted. That means if it is one atom, if it is another atom, if it is the same plane, this is the plane, then maybe the bond actually is like this. Okay, if the bond maybe is actually like this. So maybe this atom may be tilted something like this. Okay, that means this is the actual plane, but the bond is tilted like this. Then if it is the second atom, then this is the normal plane between this, but maybe the bond is tilted like this here. So the second atom may be gone here. Okay, like slightly tilted from the same plane. So the bonds do not lie between in the same plane of the atoms, rather they are a bit twisted. Due to why they are twisted? Due to the steric interaction between the, maybe due to the steric interaction between the side chains, or the atom-atom interaction, okay, or that NHCO, there is the NHCO peptide linkage, due to the NHCO linkage interaction with the side chain. So different because protein is a very complicated loop. A bond undergoes several interactions. These interactions do not allow the bonds to stay in the same planes. So it is tilted from the same plane. And the angle of tilting, say this is the normal plane where it should lie, and now it is tilted here. So this angle, okay, is called torsional angle. This angle is called, I am writing small t. Small t means this is torsional angle. That means a bond is torsioned. A bond is twisted from its normal plane. This is called torsional angle. This is designated as phi. Okay. And what is dihedral angle? Dihedral angle is the plane, is the angle between two planes. Dihedral angle is the angle between two planes. Now we are not coming to bonds only, we are coming to planes. How? Say now you have to consider two planes. Say this is one plane. To draw one plane, you need at least 2 points or more than 2 points. So say this is the one plane and say this is another plane. So these two planes are not linear. That means these two planes are not like this. These two planes are not like this. Okay. Instead of that, these two planes are like this. This and this. So the angle between the two planes, is called dihedral angle. It is called dihedral angle. Dihedral angle, which is designated as psi. This is designated as psi. Now, in protein chemistry, in protein structure, these two angles has immense importance because these two angles define the actual 3D conformation of protein. How? I am drawing a simple thing to make you understand how. So I can write the protein chain like this. I can write the protein chain like this, say NH2. One side write the protein chain, you can understand NH2, then CH2, or instead of CH2 I write RCH because this is the side chain. This may vary from amino acid to amino acid. Then CO, then NH, then again your CH. Okay, so this is R1. So here now it come R2, the second side chain. Okay, then CO. You see this, if oxygen one direction is here, the oxygen I am putting one direction is there because it is twisted. Okay, then NH. Okay, this is NH. Then CH. Again here is R3. Then this is R3. Then there is CO. Like this it will go on. This is the normal writing of a protein chain, peptide chain or protein chain, what you see. Now this is functional group CO, or CONH is the functional group. You can say this CO this amide linkage is the functional group. So this is alpha carbon, this is alpha carbon. Why is this alpha carbon? Because alpha carbon is designed the next carbon to the functional group. So this is the CONH is taken as a functional group, or you can say CO is also functional group, whatever you say. So this is alpha carbon adjacent to this CO. This is alpha carbon adjacent to this CO. This is alpha carbon adjacent to this CO. Okay. Now I have already defined you that phi angle and psi angle. Okay. Now this angle in NH2 alpha carbon and CO, that means this angle NH2 alpha carbon and CO, this angle is actually your phi angle. NH2 alpha carbon CO, this is a generalized protein chain, so I can describe any protein chain like this. So you can get this bond for any protein chain. For any protein chain, you can get this bond. So this angle NHC and C means alpha carbon and CO. This gives you a torsional angle. The bond torsion depends on this angle. We write like this: N, then C alpha, and then CO. The bond torsion greatly depends on this because this nitrogen has lone pair and this oxygen has lone pair. So this lone pair interaction do not allow the bond to be in one plane, rather to be twisted. So this lone pair interaction greatly influences the bond torsion. That's why it's called the torsional angle or phi. Next, if you come to this second part now, that is C alpha. Here you come C alpha, this angle, this C alpha, this C and this N. C alpha, C, N. Here I am starting from N, here I am starting from C alpha. So you automatically get C alpha, then there should be CO, and then the amide NH will come. So this C alpha, C, N, that means this angle is your psi, that means your dihedral angle. Okay, because this angle, here you see, it's not about lone pairs, but it is about two portion of the planes. One portion is this portion, one portion is this portion. CONH is on one portion, the polar one, and this is the non-polar one. CH is the non-polar one. So this roughly divides the protein in two planes. So among these two planes, the angle is psi, that is the dihedral angle. So hereby we can define phi and psi. Now, what is its importance in protein structure? This phi and psi rotation are restricted. This phi and psi rotation are restricted. That means they will be twisted, they will be going away from each other, or they will be going at a certain angle from the plane, from the normal plane, but they cannot move anywhere. They cannot move anywhere or to any extent. There is a limit in which direction they will move. There is the limit up to which angle they will move. This is one very important aspect of a particular protein structure. And how much they will move or in which direction they will move, okay, it will depend on what? It will basically depend on this side chain. That is R2, R1, R3. This is the side chain. Because side chain produces a lot of interactions, steric interactions, hydrophobic interactions, and even it is containing hydroxyl group or arginine group. They can even produce the ionic interactions as well, or polar interactions as well. So this side chain, because CONH or alpha carbon, this is common for every protein. But this phi and psi is not common for every protein. Every protein, this phi psi varies. For every protein, this phi and psi varies. Why it varies? The main backbone is common, but why it still varies? Because of the interaction of the side chain. Okay, so side chain and the conformation of the side chain, its loop or its linearity greatly governs this phi and psi, how much they will rotate, in which direction they will rotate. So this is called restricted, and this restricted rotation is a hallmark of a protein chain chemistry, or in broader view of a protein structure. Is a hallmark of a protein structure. Okay. Now this thing was first shown by scientist Ramachandran. Scientist Ramachandran first have depicted this unique characteristics of the protein. Ramachandran. And he has also told one thing, that depending on phi, psi can be defined. Depending on phi, psi can be defined, or vice versa. That means depending on psi, phi also can define. That means phi and psi has mutual correlationship. That means if the phi is minus 20 degree, we can predict how much psi will be. Or if there is psi, say I know the psi is say plus 60 degree, I can predict how much phi will be. So phi and psi are mutually constrained. It is called mutually constrained. Mutually constrained. Okay. This mutually constrainment is the hallmark of protein chemistry. That means if phi goes to some distance, psi who should remain within some distance. If phi goes to some distance, psi have to belong up to some distance. Apart from that, it cannot go anywhere. Or vice versa, if psi goes to some point, then phi has to restrict itself up to some point. Beyond that, it cannot go. Because if this rule is violated, if phi and psi can move anywhere, breaking their mutually constrainment, the protein chain will break. If phi and psi, if you take a spring and if you rotate the spring or you twist the spring, if you twist you try with two hands. In the left hand, you push the spring. You push the spring in clockwise direction, and in the right hand, you push the spring in anti-clockwise direction. You see, if your left hand push the spring to some extent, the right hand, you have to keep the spring rotation into some distance. If you push the spring in the right hand, beyond that distance, the spring will break. Something like that here. So this is called phi-psi correlationship. Scientist Ramachandran has correlated this thing by a plot called, Ramachandran plot. We'll come to this later. We will come to this later, this Ramachandran plot. But this is the concept of this Ramachandran plot and the basic. This is a very, very, very important aspect of phi psi. Okay. And one thing I also should mention, that if you look at the whole thing, say from this C alpha, okay, then CO, then N, then again C alpha. Sorry, not here. This up to this C alpha, then N, and up to this C alpha. So from one C alpha to another C alpha, including the side chain and the intermediate peptide linkage. This is also not linear. This is also a somewhat twisted. This portion, this linkage is also somewhat twisted. So it also produces some angle. This is called omega. This angle is called omega. So we know three things: phi, psi, and omega, which defines a 3D conformation of a protein chain, or rather the whole protein structure. The protein structure has different chains. So if you put the phi, psi, omega values for all the chains, you will get the whole phi, psi of the protein structure. So these things are very important, and among these three, this phi and psi are extremely important. Okay, this is one thing. And the second thing, this is one thing we should know. And then another thing we should understand, that we know alpha helix now. Okay, we know alpha helix. Now you know there are two types of alpha helix. One is called alpha R and one is called alpha L. What is alpha R and alpha L? The alpha R means right-handed alpha helix, and the alpha L means left-handed alpha helix. Okay, left-handed alpha helix means what the alpha helix, the screwing turn is on the left-hand thumb direction, and right-hand alpha helix means where the screwing turn is in the right-hand thumb direction. In nature, left-handed alpha helix is very rare. Left-hand alpha helix is very rare. It is there, but it's rare very. The most alpha helix we get is the right-handed alpha helix. We have to use this information when we are going to validate our computational prediction of proteins. Okay. Second thing is that usually if one alpha helix is there, it is something like this. If you take a loop, it is one loop, alpha helical one loop. Usually there are 3.6. Usually there are 3.6 residue per loop. Per alpha helical loop, there are 3.6 residues. Okay, it is approximately 4. So if one residue is here, the fourth residue is found here. So the loop is stabilized by the hydrogen bond between the first and the fourth residue. So it is written like this. This is I and I plus 4 residue stabilized conformation. If this is the ith residue, so this is the I plus 4th residue, because this is the fourth residue after this. Fourth residue after this is I plus 4 residue. So I and I plus 4 residue usually stabilizes alpha helix. Normal alpha helix stabilized by I and I plus 4 stabilized conformation. But there are some tight alpha helix. There are some tight alpha helix where where there are 3 residues, not 4. There are 3 residues in the intermediate loop. 3 residues. Okay, that means what? It is more tight. Within the 3 residues, the loop is coming instead of the 4 residues. So it is called I and I plus 3 conformation. I and I plus 3 conformation is tighter than this I plus I4 conformation. Also, there are some conformation available, which is I and I plus 5 conformation. What is I plus 5 conformation? There are 5 residues in the loop. In the I plus 5 conformation, there is 5 residue in the loop. That means I and I plus 5 loop, there is I and I plus 5th loop stabilizes the helix by hydrogen bond. So there are 5 loops in 5 residues in the loop. That means what? The residue, the loop is relaxed. I plus 5 loop is relaxed than I plus 4 because more residues are there. So more relaxing space is there. Okay, this I and I plus 3 loop is also called your 3 by 10 loop. 3 by 10. It is called 3 by 10 alpha helix because there are 3 residues per turn. There are 3 residues per turn. So it is called 3 by 10 alpha helix, and I plus 5 alpha helix is also called pi alpha helix. Pi, I plus, if this is I plus 5 alpha helix, then it is called pi alpha helix. So pi alpha helix is a bit relaxed, but 3 by 10 alpha helix is a bit tight alpha helix. So all these uh, loops, all these kind of loops have some phi and psi angles special, and that you need to be vigilant in that you have to look when you are predicting your protein structures. So these things we should know. Okay. Uh, so this is the first introduction of our protein structure. Okay. In the next class, we will move to the Ramachandran. We will move to the Ramachandran plot because that is a very important plot. Okay, one thing I have to say here, that is the beta. Okay, beta loop. There is also one thing called beta turn. One is called beta turn. Okay, beta turn. Beta turn means when a beta sheet is there like this, when a beta sheet is there, if it is turned about 180 degree, it is about turned about 180 degree. Okay, one loop to other loop, it is, if the turn is 180 degree, then it is called as a beta turn. Okay, so these are the primary notion of the protein structure. Next